Hello and welcome back. Today we are starting our three-part journey into our fourth and final macromolecule, which is nucleic acid. Today we are going to tackle a very brief overview of the structure of DNA and RNA. There's a whole nother chapter dedicated to nucleic acid, so we'll go into much more detail there, but this is just like the beginning of our understanding of nucleic acids. So let's start with the basics. It is the genetic material of the cell composed of nucleotides. So nucleic acids is the macromolecule. The monomer is a nucleotide. There are three components of a nucleotide. There is a 5-carbon pentose sugar. There is a phosphate group attached to the 5-carbon, which you'll see in the next slide or two. I don't know which one, but there's a phosphate group connected to the 5-carbon. And then there's a nitrogenous base connected to the 1-carbon. Again, this will make sense when you see the numbers in here. But again, three basic components: a sugar, a phosphate group, and a nitrogenous base. Now, there are two types of nucleic acid. There's DNA and there's RNA. DNA is probably what you're more familiar with. It stands for deoxyribonucleic acid. So deoxyribo is the sugar. Nucleic acid obviously is a nucleic acid, so basically it means that it's a nucleic acid made up of deoxyribo sugar. And then for RNA, it's a nucleic acid made up of ribose sugar. So ribo is where we get the R for RNA, and it's also a type of sugar. Now DNA is more stable and stores the genetic blueprint for cells. That's where all of our information is, and has the bases adenine, guanine, cytosine, and thymine. Typically, you see these abbreviated as A, G, C, and T. It's double-stranded and forms a double helix, and like I said, has the sugar deoxyribose. RNA is more versatile, transfers genetic information for decoding. This is what we use to transfer our DNA out on mRNA through the nucleus to the ribosome. Has almost all the same bases, except instead of thymine, it has uracil. It's single-stranded, and like I said, has the sugar ribose. Here's your main differences, right? We have a double helix for DNA. We have a single strand for RNA. Adenine's the same, guanine's the same, cytosine's the same, but thymine is only in DNA and uracil only in RNA. Here is your breakdown, and as I mentioned earlier, there was a 5' and a 1' carbon. This is what I meant by that. So on our sugar, in the nucleotide, this is on both DNA and RNA, you have the 1' sugar, and it will always count this way. The 1' is where the nitrogenous base is connected to, and then you count counterclockwise. So 1, 2, 3, 4, 5 is up here, and 5 connects to the phosphate group. 3 is down here. This is where another nucleotide could connect to if we had another phosphate, right? You could have another one. When we get into this, we're going to be saying a lot 5' to 3'. That is in relation to the sugar, the carbons in the sugar, and so we will read things from the 5' end to the 3' end. So in this case, we would be reading from like top to bottom if there were multiple nucleotides here. This 1, 2, 3, 4, 5 carbon setup is the same in every single nucleotide, so once you know it, you know it. The main difference between RNA and DNA, and you've probably seen it by now with all my talking, is that on the 2' carbon, you have an OH in RNA, and you have just an H in DNA. So the structure, okay, the phosphate group of one nucleotide attaches to the sugar of another. This creates a phosphodiester bond. Water is a byproduct, just like it almost always is in condensation reactions. And then this will form a long polynucleotide strand. Now, remember I was saying it goes in 5' to 3'? Well, this is what I meant. So 5', phosphate, sugar, 3', 5', 3', 5', 3'. So when these connect, it's 5' to 3', and then 5' to 3', 5' to 3'. So as we are going down and seeing this, we would be able to read these nucleotides in the proper order. And when I say proper order, I mean whether they're cytosine, adenine, guanine, thymine, whatever it is that the nucleotides are representing, right, whatever that nitrogenous base is. So those chains of polynucleotides are held together with hydrogen bonds. So in between your nitrogenous bases, you have hydrogen bonds. So between C and G, you have 3 bonds. Between A and T, you have 2. And you can see these as the dotted lines here. So in order for these 2 strands to pair up, though, they run anti-parallel. Do you see how in the left side here, you have your like free phosphate at the top, but here you have it at the bottom? That's because you'll notice that these sugars are facing the opposite ways. They run anti-parallel to each other, okay? So again, every time you see a DNA strand like this, the 2 strands themselves are running anti-parallel to each other. Continuing on, a little bit more specific about the nitrogenous bases is whether there are 2-ring or single-ring molecules. So you'll notice that guanine and adenine, these are double-ringed. They are called purines. And then cytosine, thymine, and uracil, they are single-ringed, and so we call these pyrimidines. I do not have a good way to remember this, except that pyrimidines sort of sounds like pyramid, and pyramids have a single point, and so there's only 1 ring. I don't know if it's super helpful, but that's the best I have to remember it, but I don't really also have a good way to remember which are which, so this might just be straight up memorization. So moving out of like the basics and into specific types of RNA, there are 3 types that you will be responsible for knowing. There's messenger, abbreviated mRNA, transfer, abbreviated tRNA, and ribosomal, abbreviated rRNA. Messenger is a copy of a gene which encodes a specific polypeptide. So it's this mRNA copies a gene of DNA, and it will be used as the template for creating a protein. tRNA carries the polypeptide subunits, or amino acids, to the ribosome to continue on in making a protein. So mRNA has the template, tRNA brings the amino acids, and then rRNA is just a primary component of the ribosome, and it's responsible for like its enzyme activity, its catalytic activity. Now for our last little bit about this, we're going to talk about some of the scientists and some of the major people responsible for DNA's discovery. So Watson and Crick are probably your most famous people, but there's also Franklin and Wilkins. And I know, I have my fingers crossed right now. I'm hopeful that you know who Franklin is. Rosalind Franklin. She was a woman scientist during this time. She was instrumental to discovering the shape of DNA. And Wilkins was her partner. Maurice Wilkins was her partner in all of this, but Franklin is the one that kind of got the short end of the stick here. So let's go through some of this. So Watson and Crick proposed the correct structural organization of DNA in 1953. I know that you're probably thinking 1953 is like so long ago, but it's really not. It's like 70 years ago, which, yes, to be fair, that is a lot of time. But in the grand scheme of things, that is like a blink of an eye. We have only known the basics of DNA for 70 years. That is what all of our body uses to create all of our traits, all of our proteins. Everything that our body does relies on DNA, and we didn't even know what it looked like until 70 years ago. I find that just incredible. We've leaps and bounds done so much with that information in the last 70 years. It's truly incredible. Now, back to the history of this. Watson and Crick based their findings on really key experimental discoveries. So 3 specific discoveries. Phoebus Levine, was responsible for determining that nucleotides are made up of a sugar, phosphate, and a base, which we know is true. Edwin Chargaff determined that there are an equal number of purines and pyrimidines, which means that if you have an A, you must have a T. If you have a C, you must have a G. They have to be together. There are an equal number of the 2 of them. And then, of course, Rosalind Franklin determined that DNA was organized into a helical structure. So using trial and error, Watson and Crick were able to assemble the DNA and determined that there was an anti-parallel double helix, complementary base pairings, and outer edges remain exposed, which allows access to replication and transcriptional proteins. Basically what that means is because the outer edges are exposed, that means that DNA can replicate and it can go through protein synthesis. Now, early models of DNA thought that it was a triple helix, meaning that there were 3 strands. The bases were on the outside instead of the inside, and that there was not complementary base pairing, meaning that A could match up with whoever was there. It didn't have to be a T. Now, why am I bringing all this up about Franklin at the beginning? Well, because the final construction of DNA that Watson and Crick so famously got credit for was almost exclusively relied on the crystallography data that Rosalind Franklin produced. So Rosalind Franklin was the first to actually take this image that you see right here and determine that it was a double helix. So she was the one that confirmed that DNA was arranged in a helical structure, and the issue is that this data that she found was shared to Watson and Crick without her knowledge or permission, and Watson and Crick took it and published it. So they got the credit for her work, and what the worst part about all of this is eventually they went on to win the Nobel Prize for DNA, for determining the structure of DNA. And so Watson and Crick and Wilkins, Franklin's partner, did get the Nobel Prize. Now you're probably like, whoa, was it because she's a woman? And unfortunately, well, because most women scientists did get excluded strictly because they are a woman, but Rosalind Franklin unfortunately passed away due to cancer a few years before the Nobel Prize was awarded, and you cannot award Nobel Prizes posthumously, so she went on without the winning of the Nobel Prize. But her work is instrumental to the discovery of DNA. So hopefully, if you didn't know who Franklin was, you now know who she is, and you can thank her for figuring out the shape and the model of DNA. Great job, and I'll see you guys in class.